Hello, everyone. My name is Ferralita-Madere, and I'd like to thank the organizers of this conference for the chance to talk about some really exciting work studying the Transkingdom connections in the female reproductive tract and health and bacterial vaginosis. Within the female reproductive tract, or FRT microbiome, the bacteriome has been extensively studied. Unlike in other mucosal microbiomes such as the gut, FRT health is associated with low-diversity bacteriomes dominated by lactobacillus species, which confer health by producing lactic acid, stimulating the production of anti-inflammatory cytokines, and directly killing pathogenic bacteria. Conversely, high-diversity bacteriomes composed of facultative and strict anaerobes, such as Gardnerella, Prevotella, and Sneathia, are associated with bacterial vaginosis and poor female reproductive health outcomes. Preliminary investigation of the FRT virome has focused primarily on the DNA eukaryotic virome. Bacteriophage are viruses that infect bacteria, make up the phagome, and are the largest and most abundant viral members capable of modulating bacterium composition through lysis. Due to the prey and predator relationship between bacteria and bacteriophage, it's likely that these microbes participate in transkingdom interactions dependent on the disease state of their environment. This leads to our interest in studying these connections within the FRT. Globally, bacterial vaginosis, or BV, poses a significant threat to female reproductive health. BV is characterized by a shift in vaginal microbiota away from lactobacillus dominance towards a more diverse collection of facultative and strict anaerobes. Numerous studies have linked BV to an increased risk of STI acquisition and preterm birth, highlighting its impact on the FRT and the need of investigation of microbial members implicated in this disease. With this, we have formed our hypotheses that bacteriome shifts seen in urogenital diseases such as BV are associated with specific FRT phagome, and that signature bacteria and bacteriophage associations are indicative of health or disease. To address this hypothesis, we used a cohort of 50 HIV positive and 50 HIV negative young sexually active South African women that were recruited as a part of the University of Cape Town HIV-HPV study. Vaginal swabs were self-collected at 6-month intervals, and pap smears were performed annually. In this study, we focused on BV, which was diagnosed clinically, finding 54 BV positive and 46 BV negative participants at baseline. Our first goal of this study was to characterize the microbiome within the FRT and identify microbial signatures of BV. 16S rRNA gene profiling of the V3-V4 region was performed on total nucleic acid extracted from vaginal swabs. Wards linkage hierarchical clustering grouped compositionally similar samples into distinct bacterial community profiles called community groups, or CGs. Through this, we identified 5 major CGs distinguishable by lactobacillus dominance, CG1 or CG2, which were made up of Lactobacillus crispatus and Lactobacillus iners, respectively, or high-diversity polymicrobial CGs 3 through 5, notably including Gardnerella, Prevotella, Sneathia, and Shuttleworthia, among many other taxa. We also found that CGs 3 through 5 exhibited significantly higher alpha diversity compared to CGs 1 and 2 and correlated positively with BV. With our viral analysis, we focused on baseline samples reflecting 14 BV negative and 24 BV positive women. Sequences from over 492 known bacteriophage species were detected within the vaginal samples. Neither VST exhibited a dominant bacteriophage member, with both comprising diverse viral taxa. Like our work with the FRT bacteriome, we explored the phagome and investigated bacteriophages also clustered into distinct communities. Interestingly, we are the first to describe bacteriophage community groups based on composition and abundance, termed viral state types, or VSTs. Here, we see that bacteriophage Shannon diversity differed between VSTs, with VST2 having the highest diversity bacteriophage population. This led us to question how bacterial CGs and VSTs are linked. If you look here at the top, you'll see viral state type and community group designations for each sample. Of note, the VSTs significantly correlated with bacterial CGs, with VST1 associating with CG1 and 2, the lactobacillus dominant groups. We also see that VST2 associated with CD3 and 4, which were both high diversity CGs. Next, we want to investigate transkingdom associations between bacteria and bacteriophage that could suggest possible interactions associated with BV. Kendall's correlation analysis showed that Bacillus virus camphawk and pony, which both infect Bacillus genus bacteria, positively correlated with BV-associated bacteria Gardnerella, A. vaginae, Prevotella, Sneathia, and Dialister. Reads assigned to unclassified E12 bacteriophage also positively correlated with Gardnerella and A. vaginae. Excitingly, some of these same bacillus-infecting phage, including Bacillus virus pony and Staley, were inversely associated with bacteria protective of BV, particularly Lactobacillus iners. From this work, we can conclude that much like bacteria within the FRT, bacteriophage communities cluster into distinct VSTs that differ in abundance, composition, and diversity. These VSTs are significantly correlated with either health-associated bacteriomes dominated by lactobacillus or polymicrobial bacteriomes associated with BV. Distinct bacteriophage taxa were found to strongly correlate with bacteria protective or indicative of BV. And with this, these data together suggest that bacteriophage could possibly be modulating bacterial populations in a transkingdom manner to promote diseases such as BV. With that, I would like to greatly thank both present and past members of the Monaco Lab, my collaborators on this project from both URMC and the University of Cape Town, and my funding sources. Thank you.